Okay, good morning, everyone. Some reason I always feel like I'm last. Okay. So I'm going to talk to you about a phase 1 trial that we did with the other anti-CD38 antibody. In fact, there's 3 in development. There's one, daratumumab, that you heard about. I'm going to talk about SAR650984. I'll call it SAR from now on. And there's another one also from Celgene, the MOR202 drug. Now, these all bind to a different part of the anti-CD38 receptor, but whether that makes any clinical difference, we certainly don't know at this point in time. I want to set the stage by saying that, there is an unmet medical need in myeloma. And the unmet medical need is that really we have limited, what I call blockbuster agents in myeloma, meaning agents as a single agent have significant activity in the relapse and refractory setting. What we have is IMiDs. You may know of some of these drugs. The IMiDs are thalidomide, lenalidomide, otherwise known as Revlimid, or pomalidomide, otherwise known as Pomalyst. Those are the IMiDs that are blockbuster drugs. And then we have the proteasome inhibitors, bortezomib or Velcade, or Kyprolis or carfilzomib. And with those 2 blockbuster classes, we've really advanced the overall survival for patients with myeloma. We've changed it from potentially a 3-year survival that hopefully now is more like a 7 to 10-year survival. So that's great. I'm going to talk to you about a more heavily pretreated patient population. And in these patients, the Mayo Clinic, Keith's colleagues and himself have published extensively on this. The average survival really is about 9 months in the patient population I'm going to talk about. And I'm going to tell you that these CD38 antibodies, I think, are blockbuster drugs. These are real drugs. We should be in here talking about CD38 because it's an important component to myeloma therapy. Now, as Maria said, the way these antibodies work, they bind to the surface of the cell and they hold up a flag. And they're simple. They're naked antibodies. All they're doing is holding up this flag. It's different than what you heard about with brentuximab that takes a toxic payload to the drug. This is just putting a flag on the cell and saying, immune system, come get me, okay? And it's worked very well with drugs like rituximab in lymphoma, and so in myeloma, we're, I think, way behind. We're 10 years behind lymphoma with the use of these antibodies, but we finally have some that actually work. Now, in our study, we combined it with lenalidomide, and lenalidomide, it has a lot of, I told you, it has a lot of anti-myeloma effect, but the one way it has an anti-myeloma effect is that it actually stimulates the immune system. So it tells the immune system, hey, go look for these flags. And so the reason we put this combination together is we thought that the antibody itself would work better if we combine it with lenalidomide, or an immunostimulatory drug. Now, I don't have the data to show you here, but we've used these anti-CD38 antibodies as a single agent in a similar population and found an overall response rate of about 32%. Okay, and that's that's the background for this study. So this study is a phase 1 study. The goals of a phase 1 study are actually to look at toxicity. Is the combination safe? And this included adults with relapse and refractory myeloma. They had to have at least 2 prior therapies, and prior autologous transplant was fine. There was actually no upper limit on the number of therapies. So in fact, this ended up being, a very heavily pretreated population. And before they start, they just had to have adequate bone marrow reserve. I hope you guys can all see the color of these slides. In any event, the way we did this study is we did a standard what's called a 3+3 design. You put 3 patients on the lowest cohort. The lowest cohort was the SAR drug at 3 milligrams per kilogram given every other week with standard doses of lenalidomide and dexamethasone. You enroll 3. If they do fine, then you go up to the next cohort. So we went from 3 milligrams per kilogram of SAR every other week to 6 milligrams to 10 milligrams of every other week. And in fact, it was very well tolerated. In the combination, there were no unexpected toxicities. And SAR is and these antibodies tend to be very well tolerated drugs. There is an incidence of infusion reactions, and I think I have a slide about that in a second. Now, once you get to what was our top dose, the 10 milligrams per kilogram, and it was safe, Then we enroll in what's called an expansion cohort, more patients, to see if we can get some preliminary evidence about efficacy. So here's the patient population. I'll show you that overall, there were a heavily pretreated population. Now, we divide myeloma therapy into regimens, meaning, they got this double therapy and this triple therapy, and also lines of therapy. The difference between regimens is lines is you may get one regimen as induction and knock the myeloma down, and then a transplant to further knock the myeloma down, and then maintenance to further knock it down. That's 3 regimens, but it's only one line of therapy. Line of therapy means it's gone down, and then they've relapsed. And then they've got another line of therapy to make it go down again. So on average, they had 4 lines of therapy. So in the Aspire trial, it was 1 to 3 lines of therapy. So it was a much less heavily pretreated population. Now, if we look at the characteristics of the therapies these people received prior, more than 90% received prior lenalidomide, more than 90% received prior bortezomib, about a third of the patients had prior pomalidomide, and almost 50% had prior carfilzomib. So many of these patients were what we consider double refractory or refractory to our our most potent blockbuster drugs. And in fact, in terms of the IMiDs, you can see in the red box, 84% of the patients were relapsed and refractory to their last IMiD-containing regimen. Okay, so I told you that the SAR drug is very well tolerated. The only side effects really seen were infusion reactions. These occur about a third of the time. They're mostly mild, grade 1 and 2, meaning the person gets a little rash or gets a little itch or gets stuffy nose. We only had to actually stop the infusion in very few, like 3% of the infusions of the overall infusions throughout the whole study, do we have to stop the infusion and then restart the infusion. And then after the second cycle of this therapy, there are no infusion reactions. So it happens in the first couple cycles, and then it's really not a toxicity that we see long term. So what about the responses? So if we look at the overall response rate in the 31 patients that were treated, the overall response rate was 58%, with a clinical benefit rate, meaning adding the minor responses, of 65%. If we look at just the patients that had the 10 milligram per kilogram dosing, 24 of the patients, this was basically the highest dose of the antibody, we see basically an overall response rate of 63%, double what we saw as a single agent, and a clinical benefit rate of 67%. And we did have 2 stringent CRs in this patient population. Now here's the waterfall plot. Now I don't know if you guys are used to seeing waterfall plots. This is the waterfall plot. Below the line means they've had a reduction in their M protein. This is a dramatic waterfall plot. You can see that the majority of these patients had some response. And again, 84% of these patients were refractory to IMiD-based therapy. So if we just gave them lenalidomide, these should all be above the line. So this combination was very, very active. Now if we look at progression-free survival, in this small cohort of patients, the progression-free survival is about 6.2 months. So I'm going to simply conclude by saying that this combination was well tolerated. There were no unexpected toxicities, and in my mind had a fairly dramatic response, with 2/3 of the patients having a response even though they were refractory to our other blockbuster agents. And lastly, I do think these anti-CD38 antibodies, these are the next blockbuster class of agents. These are the next agents that are really going to show some benefit to myeloma patients, and the next 5 years is going to be really fun, moving them from the refractory setting to the less refractory setting to the frontline setting. It's going to be great. Thank you very much.
Thank you, Dr. Martin.